Okay, hi, my name's Tom. I'm currently on the MRes and Synthetic Biology course here at UCL. And as Philip indicated, I'm here to talk about a couple of things. What synthetic biology specifically is, what you can expect out of the MRes course, perhaps a couple of things that interest me specifically. Okay, so I left industry about 2 months ago. One of the hardest things I had to do was explain to my then boss what I was leaving to do. So I'm hoping that you guys won't hit this problem. So what is synthetic biology? In a nutshell, synthetic biology is an approach to engineering biology. So that means that specifically it's not metabolic engineering. It's not bioprocess engineering. What it is an approach to do these kinds of things. So another way of saying this, and I love this quote if I get this down a bit, is synthetic biology isn't about what you're making, it's about how you're making it. So let's look for a second about fundamental principles of synthetic biology. So if we look back at the last 30 years of biotechnology, we can see that it's based on 3 fundamental principles. The first one of which is the use of recombinant DNA, so sticking bits of DNA together, right? Ligation reactions, that kind of thing. The second thing is PCR, the ability to take specific sequences of DNA and amplify them up. And the third one is automatic sequencing. So essentially what you've got here is the first 2 are writing DNA and the third one is reading it. And altogether that's genetic engineering. So how is synthetic biology different? Well, synthetic biology builds upon these core principles to create something new by introducing 3 new fundamental principles. The first one of which is the automated construction of DNA. So the genetic engineer was using recombinant DNA for this. So what was he doing? Well, if, let's say, a genetic engineer wanted to investigate a gene of interest, well, what he's got to do is he's first got to isolate a plasmid, cut that plasmid with an enzyme, a restriction enzyme. He's got to find his cell line with a gene of interest, extract the DNA, break that up using the restriction enzyme again, hybridize the 2, and then he's got a library of different plasmids with different genes in them. He's got to select which one's got his gene of interest. That's recombinant DNA, simply. And that's a lot of work. The synthetic biologist looks at this and goes, nah, not interested, just get me this DNA. Literally emails a sequence to a company to print it. So, well, how does that company do that? Well, they use one of these things. This is a DNA printer. It works almost exactly like your printer at home. And it goes backwards and forwards and it spits base pair, base pair, base pair, base pair until you've got the sequence that you require. And if you look at the top, you'll see some bottles, and literally 4 of those will be labelled A, T, C, and G for the 4 bases. So the second fundamental principle behind synthetic biology is that of standards. What do we mean by standards? Well, I'm going to introduce the concept of biobricks here. I'm not going to go particularly too deep into them because you'll have the iGEM team and Darren Nesbitt over there who will talk to you a bit more specifically about them. But essentially, biobricks are standard biological parts. So I'm sure you've heard of things like promoters, specific genes, things like that. That's what comprises biobricks. And they contain DNA sequences of specific structure and function. So the idea behind this is I could say to Philip or Darren, I want a biobrick that enables me to survive salt toxic conditions. And he could go, there you go. And I could plug it into my host organism. And that's actually the final point. It's designed to be incorporated into a host chassis, whichever one we might like to. So the third fundamental principle is that of abstraction, and this comes kind of from computer science. So when I got the email about this talk from Philip, he sent me an email using text, right? He used an email program that was no doubt written in C++ or equivalent, and that was actually executed by the computer on a binary level. Now Philip interacted with the top level of this. He didn't need to know about C++, I'm sure he does. He doesn't need to know how to code in binary, although I'm sure he does. So really what we want to do is we want to interact with the highest level as possible. And the same is true of synthetic biology. We want to interact at this devices level, not necessarily have to worry about the specific sequence involved or the parts, but we want to be able to do this, well, simply. So, for example, if I wanted a toggle switch, I could approach Darren Nesbitt over there or Philip. I'd say, I want a toggle switch. And he'd be able to give me one. I wouldn't need to know what biobricks are involved or what specific sequences are involved. In fact, actually, I wouldn't necessarily need to know that DNA had 4 different bases. I could just plug it in and work. and it would work. That's the general idea. We're not quite there in terms of synthetic biology quite yet, but that's where we're trying to get to. So, looking over our definition, synthetic biology builds upon the core principles of genetic engineering. Recombinant DNA, PCR, both used to write DNA, and automated sequence to read it. And then introducing these three new principles of automated construction of DNA, standards, and then abstraction. Okay, so if you can take one definition away from this talk, it's this. Synthetic biology isn't about what you're making, it's about how you're making it. Love this quote. Okay, so I just want to briefly touch upon the Masters in Research in Synthetic Biology. I started it about a month and a half ago, back in September. It's split into 3 taught modules, 2 general research skill modules, 1 synthetic biology taught module, and the 8 to 9 month research project where you can do just about anything. It's an interdisciplinary thing. And about 2/3 of the marks are located in that research project. So the person to email is John Ward about this if you're interested. So just as a last wrapping up discussion, I think I'm almost just about out of time. I just want to introduce 2 interesting things. Usually in these kind of talks, this is the point where I talk about something that was awesome and going to happen in 10, 15 years' time, possibly, maybe, someday. But I'm going to introduce 2 things that happened this year that really excite me. The first one is non-volatile data storage encoded at a genetic level. So what this means is I can store data in bacteria. So I'm not saying that I can store this whole presentation. Just 1 bit is what I can do at the moment. So the advantage of this is I can create, well, potentially in the future, biocomputers. So what's exciting about that isn't so much the fact that biocomputers are going to overtake these kinds of computers. What's exciting about that is that I can potentially take computers into areas where silicon computers, conventional computers, can't go. And if you're interested, this came out of Stanford Lab about a couple of months ago. And the other one is tumor-targeted bacteria. These things are awesome. We're looking at a potential cancer treatment. What these do is you essentially introduce bacteria into your system. They isolate them, they localize themselves to the tumor and start expressing tumor factors. So you've got a highly targeted response. And I'm introducing these 2 systems twofold, for 2-fold reasons. A, they're both pretty awesome, and B, they're very, very different, but the concepts behind them are quite similar. Okay, so if I've got any questions on that, I'll take them. If not, I'll move on.
You said what synthetic biology could potentially be, but you also said that we're not quite there yet. What is that?
Well, realistically, what we want to do is be able to construct systems 10 or 20 genes at a time, maybe even 100. So if you wanted to create, I don't know, a whole network, you could essentially plug it in, but that whole network is going to be constructed of maybe 100 to 150 genes. We don't have the ability to construct those genes effectively at that high level at this point, but there are things that are being looked at to do this, things like normalizing prime UTRs and primer expression rates, things like that, and also normalizing gene expression on each biograde, so that kind of thing.